Good afternoon. Thank you for being there. I hope you're having a great meeting. I wanted to give you a perspective of our cell engineering approach to deliver therapeutic payloads in autoimmune diseases, what we call the gene-engineered pluripotent stem cell approach, GPS. You guessed that there'll be some sort of targeting in the approach, right? Otherwise, I wouldn't have chosen the GPS kind of name for it. So what is the challenge? There is still unmet medical need for several diseases, and there's an opportunity to have an off-the-shelf cell therapy delivering a payload therapeutic. And we aim to do that using iPSC-derived MSCs and engineer them for a targeting approach and a cytokine or other gene of interest-driven factor at the site. There is a new opportunity. We've seen a lot of development in autoimmune disease, and there will be some that will have fantastic new approaches. We believe that there's still several of them, and I'll come back to the indications, that remain an unmet medical need, and we would need an approach that can be off the shelf. And this off the shelf is based on engineering them, as I said, with a lenti approach to deliver the gene of interest and using an iPSC sourcing, really optimizing the opportunity that MSCs can deliver with optimal sourcing and manufacturing flexibility coupled with intensifying the mechanism of action. We have a couple of products in development. One that is still donor-based, Kiji 01, transfected with IL-10 CXCR4, and another one that is still in R&D stage, iPSC derived with the same proprietary bicistronic gene for IL-10 CXCR4. We have the team in place, path of the financing, and the opportunity to go to the clinic pretty quickly. And if this would move, there we go. Here is the experienced team. I'm here, my colleagues are trusting on me to deliver the message to you. And this is our current portfolio. We do have an asset ready to go to the clinic. We are in final stages of lenti GMP production. All the preclinical package is completed, and we are aiming to be in the clinic with the first patient, first visit within the first half of next year. And this will be, as I said, for graft-versus-host disease, an indication that still represents an unmet medical need, particularly for late-stage refractory patients, even though opportunities, including with MSCs, are surfacing, and so validating the opportunity on which we want to bring a step up with the engineering, with gene engineering. We have, and you will see, very attractive data, particularly on long-term and cell population modification in IBD, which believes that with adequate patient targeting, we do bring a significant value-added proposition to certain subgroups of patients in IBD. And we do have incoming collaborations with different organizations that will bring other genes of interest. Really, iPSC-derived MSCs may represent a fantastic opportunity to carry a targeted therapeutic approach, and that is the basis of what we do. The first asset, as I said, is really using that cell engineered with CXCR4 targeting inflammation, where the IL-10 is delivered in high quantities, providing an immunomodulation that is driven by the presence of IL-10. And I'll come back to some elements of that mechanism of action driving that. So if I slow down a bit and take you through the process here, currently we're sourcing from adipose tissue, a well-established European agency validated for another product sourcing of our MSCs, but we will move to iPSC-derived. we will engineer it with the CXCR4 IL-10, and we will have an off-the-shelf IV administration. This will provide a targeting to the inflammation. As you can see, inflammation includes multiple factors, including STF1, which is the connection to CXCR4. So as it increases in inflammation, with IV administrations of the cells, we get a targeting to the tissue. And once at the tissue, we do have an increased production of IL-10. It's in a way a professional, ready to act, hit the road running cell that will deliver the immunomodulatory stimulus that we know they deliver, but is now potentiated by a constitutive gene transfection. And so we have multiple evidence in terms of preclinical and also some data of in vitro migration. We have in vitro immunomodulation increased over and above non-modified MSCs. We have multiple animal models consistently showing that despite some effect of the MSCs, we have a significantly increased benefit by the transfected cells. And we have gone beyond by documenting the biodistribution, i.e. we see the transfected cells at the site of inflammation. We see our codon-optimized IL-10 at the site of inflammation being produced. And we see a switch on inflammatory cytokine profile to non-inflammatory. We see an increase of Tregs and Bregs, and very interestingly, incoming data, other populations, namely macrophages, which could explain the data on long-term benefit. There's been reports of long-term benefit in GVHD with unmodified MSCs. We believe that as we will potentiate the short-term effect, we might also have a long-term benefit increased with these transfected cells. So really optimizing the incipient effect that we've seen for some time, using state-of-the-art current technologies that enable us to so deliver. A few pieces of data. So CXCR4 does contribute to migration, and we've seen that in vitro. Increased IL-10 production gives a more potent immunomodulating stimulus, and we've compared that in vitro. In the animal model of GVHD, we have the usual benefit with the MSCs, but we increase that with the transfected cells in a humanized GVHD animal model, where we can also document the switch on the cytokine profile, as well as an increase of Tregs in circulation, Bregs and Tregs in the spleen, and as I mentioned to you, in situ myeloid cells changes as well. So overall, a mechanism of action documented at cytokine and cell level. Now, with that in mind, we are progressing towards a clinical study where it's a simple manufacturing process, as you would expect, collection or iPSC sourced. The cells will be expanded, lenti transduced, and once transfected, they will be further expanded, cryopreserved, and formulated for off-the-shelf use. They will be formulated in multiple size bags so that the patient will receive a dose without resuspension or counting. It's really just straight injection to the patient upon thawing. And the clinical study is about to go. There was a clinical study ready to be executed when we took the license of the asset, and it will be conducted in multiple sites across Spain. There will be a dose ascending, and we're targeting the dose of 1.8 to 2 times 10 to the 6 per kilogram. And you can see the 1.8 to 2 gives us the flexibility for the different doses formulations so that we make it clinically convenient. Usual endpoints with a comprehensive analysis of the biomarkers, really to document the mechanism of action and some long-term follow-up. So we believe that we come with a proposition that within the context of GVHD will target about 25% of the patients in third line, potentially progressing to second line. And we do have a scale-up program that will make the cost of goods quite attractive. We have the safety that we all have with MSCs, but we bring a very significant increase on the immunomodulatory capacity. So it's kind of taking advantage of the good work and the good benefit MSCs have delivered, but taking it to the next level by the intensity and the targeting of the immunomodulatory benefit. And you will know about the needs of the GVHD market, and I will not labor on that. But I would like to mention to you that we're going beyond that, and we have other indications, in particular IBD and solid organ transplant, as well as others on data. Very interestingly, we're seeing long-term upon re-exposure benefit in the animal model, in the DSS animal model, potentially associated with some of the myeloid cells changes that I mentioned before, which could give us an opportunity to really play a role in addressing at least a fraction of the patients with IBD. Similarly, based on the evidence of GVHD and the similarity of the mechanism of action in solid organ transplant, we may have an opportunity to target that indication. We're actually looking at collaborations with organizations that have a vested interest in this indication to really develop it as well. As an organization, we are in seed and seed extension mode, and we see ourselves delivering by second half of 25, we're successful with the seed to a iPSC-based platform of gene engineered for a purpose, together with initial clinical data, and go beyond that with the Series A for the steps that I mentioned before. So we will be addressing a refractory GVHD already with a seed, and then progress to IBD with a platform, and aim at other indications, potentially in collaboration. We have had an initial pre-seed plus non-dilutive funds. We're expanding that in currently active raising mode, like most of us all the time, to take us to the deliver that I mentioned before, and then the Series A to go beyond, and along that make discussions in business development as you would expect. And we're getting good traction outside of Europe, namely in Asia. Now, so we see ourselves as a transformational approach, taking advantage of engineering cells for a purpose, but using also an optimized IMSC sourcing. We're about to be in the clinic, and we have extensive mechanistic and in vivo preclinical data that would enable us to support our hypothesis. And so we see ourselves as a leading or potential leading organization with engineered MSCs derived from iPSC. We're trying to raise, as I mentioned, and we have an attractive name that represents the collaboration between France, where the company was founded, as well as Spain, where virtually all activity takes place and the team is based. Thank you very much. Happy to answer any questions here or outside if you so wish.